Val, simplement.
Em separo, potser, perquè així no em sentis? Hola, Pau, em sents? Pau? Bueno, pues això ens està bé, perquè així ens em pots sentir o no. Bon dia. Estic cagat. Moltes gràcies, Maxi. Bones. Molt bé. Per cert, Aina, els hi va agradar molt el brownie. Sí. No estava cremat. No.
En principi sí que funciona.
Si voleu... Yeah? Sí, ja els he tancat. Sí, només he deixat oberts els del final. Tinc l'ampolla. 2. Jo... Sí. Bueno, ya lo haremos después.
Buenos días a todos. Yo creo que se me escucha. Sí, no se escucha, me encanta. Ok. Buenos días a todos. Vamos a empezar entonces con este acto de defensa de la tesis doctoral de Oriol. Os presento a los miembros del tribunal. A mi derecha está Sergi Stals, es Associate Professor at the University of Barcelona, and he will act as secretary. On my left side is Dr. Ana Micola from Aalto University, Assistant Professor. Y yo misma soy Marta Carvalla, profesora titular del Departamento de Ingeniería Química de la Universidad de Santiago de Compostela. El acto consistirá en dos partes diferenciadas. Una primera parte en la que Oriol nos presentará los resultados más relevantes de su trabajo, que durará aproximadamente 45-50 minutos aproximadamente. Y luego pasaremos a la segunda parte, en la que será el debate con el tribunal, que esperemos que dure aproximadamente 1 hora. Vamos a intentar eso como target. Entonces, without more delay, the floor is yours. So we will move up front to follow your presentation, and then you can start.
Thank you very much, Marta. Well, welcome everybody to my PhD dissertation. The title of my thesis is The Impact of Bioreduced Graphene Oxide on the Anaerobic Transformation of Emerging Contaminants. And this thesis was carried out under the supervision of Professor Jelena Radjenovic and Dr. Maite Pijuan. I organized this thesis following the traditional organization. I will start with a short introduction, and then I will define the main objective of my thesis. And following that, I will explain briefly the methodology. Finally, I will finish presenting the results that are organized in 4 chapters. And at the end, I will present the main conclusion of my thesis. Let's start with the introduction. I want to start talking about wastewater treatment and how a wastewater treatment plant is organized. Once wastewater arrives to a wastewater treatment plant, there is a first stage that is the pre-treatment of wastewater, where the main particulate matter is removed. And there is a first clarifier, where a primary sludge is collected after the separation. Then wastewater is directed to the secondary treatment, that is a biological treatment. And the most implemented technology is the aerobic wastewater treatment. that consists in supplying huge amounts of air by pumping air. And the main characteristic is the low retention time and the fast growth of the bacteria. After this biological treatment, there is a secondary clarifier where the sludge produced in the biological treatment is separated, producing the effluent of the wastewater treatment plant that it's discharged into the environment. This secondary sludge and also the primary sludge are mixed and redirected to an aerobic treatment, an aerobic digestion treatment, where this sludge is digested and biogas is produced. The main characteristic of this treatment is that methane is produced as a final byproduct, but the feature of this bacteria and archaea is that the cellular growth is slower. So we need higher hydraulic retention time. Which are the main challenges in wastewater treatment? The first main challenge is the energy consumption. As I said, in the secondary treatment, the biological treatment, we need to pump huge amounts of air. And it's estimated that more or less the 2-3% of it represents the 2-3% of the energy national consumption in developed countries. Also, it's very important to highlight the emission of greenhouse gases. This air is used as an electron acceptor. And the final product is mainly CO2. We oxidize organic matter. And there are other greenhouse gases emissions that are being produced during this treatment. Moreover, the carbon emission neutrality, it's a very important aspect to be achieved. And this is why anaerobic digestion is used to recover methane that can be used as energy source in thermal engines. And it's considered a resource recovery strategy. But there is another challenge that is very important to be addressed, that is the presence of emerging contaminants in the wastewater treatment matrix. These emerging contaminants is a group of molecules that are very persistent, are not easily degraded by biological treatment, and pass through all the stages in wastewater treatment plants and finally are discharged into the environment and can be detected in environmental samples. The main problem of these molecules is basically the huge environmental impact, and also in the case of antibiotics, the proliferation of antibiotic-resistant bacteria. That in the case that this kind of genes ends up in a pathogen, we cannot treat a disease with the same antibiotic. Let's talk now about the microbial metabolism. The aerobic metabolism, as I said, uses oxygen as a final electron acceptor, producing CO2. And the main advantages is the reaction rate, and it's very effective removing organic matter. However, the main limitation, as I previously mentioned, is the energy consumption in the sludge production. Concerning the anaerobic metabolism, a final electron acceptor are other molecules besides oxygen, as can be sulfate, nitrate, this is in anoxic conditions, and organic molecules or CO2 in anaerobic processes, producing methane. However, we have some limitations, as I said. The reaction rate is much lower. But at the same time, we produce less sludge and we can recover energy from these processes. And this can lead to the energy neutrality of the overall process. The anaerobic digestion process, the general scheme, is that it consists of 4 main steps. The first step is considered the limiting step because of mass transfer limitation. It's called hydrolysis, where particulate organic matter is break down into monomers. After that, we have the acidogenesis and acetogenesis steps, where volatile fatty acids, long-chain fatty acids, and finally acetate are produced. The last step, the most important step, is the methanogenesis, where methane is produced and can be produced through 2 different pathways. The hydrogenotrophic pathway, where CO2 is reduced using hydrogen as electron donor, and the Wood-Lungal pathway, that is the acetoclastic pathway. reducing acetate. I previously mentioned that hydrolysis is the limitation step of this process, but if methanogenesis is inhibited, if methane is not produced, we can have a problem with the acetic acid accumulation in our reactors. This process should be carried out at pH 7.5, but if volatile fatty acids are accumulated, then the pH will decrease. And this could lead to the complete inhibition of the whole process. These different steps in anaerobic digestion needs of a consortia of bacteria and also archaea, two different kinds of microorganisms. And at the same time, we need a correct electron flux and carbon flux. What's the electron flux? Well, these microorganisms exchange electrons using basically biomolecules that are hydrogen and formate, as you can see in this scheme. However, there are other mechanisms that are biological structures that are kind of proteins that can connect cell to cell these bacteria, and they can exchange electrons. Finally, it was discovered that these bacteria can also exchange electrons through conductive materials. There are many different categories of conductive materials depending on the properties of the material. It can be methyl-based materials or carbon-based material. In my case, I use carbon-based materials. And the exchange of electrons through these conductive materials, it's called direct interspecies electron transfer. In my thesis, I studied graphene oxide, but I want to start talking about graphene, that it's we can call it the parent compound, the parent material. This graphene is a monolayer of carbon with superconductivity properties, but it's very expensive to produce. On the other side, the material that I use, that is graphene oxide, is non-conductive, but it's very similar to graphene oxide, but contains a lot of oxygen functional groups. It was discovered as well that if this structure is reduced, these oxygen functional groups are removed, we can achieve similar properties than graphene. And the main reduction processes are the biological reduction process that is used in microorganisms that use these oxygen functional groups as electron acceptor. So at the end, we obtain bioreduced graphene oxide. There is the hydrothermal processes applying huge amount of temperature and pressure, but it's very costly. And finally, there is also the chemical reduction using reagents. But at the same time, this option produces a lot of waste. For this reason, I basically studied the biological reduction of graphene oxide. So the main objective of my thesis were to investigate the impact of graphene oxide on the performance of anaerobic bioreactors to enhance the yields and the rates in anaerobic digestion process, and also to investigate the removal of emerging contaminants during anaerobic treatment in the presence of graphene oxide, and try to determine the associated removal mechanisms. This is an overview of the methodology I used. During my thesis, I operate 3 different kinds of biological reactors. I quantify pharmaceuticals using analytical chemistry techniques as SPE for concentration, and also target and non-target analysis. And finally, the DNA and RNA sequencing were carried out using the sequencing the 16S gene and also mRNA sequencing. To go a little bit more in deep in the anaerobic bioreactors operation, first of all, I want to talk about batch and fed batch reactors that are basically a mix of graphene oxide, substrate, and inoculum. The inoculum is anaerobic sludge from the wastewater treatment plant. I was dosing graphene according to the volatile solids concentration in the inoculum, and the substrate was dosed following a ratio of 2 with the inoculum volatile solids as well. Then the difference between batch and fed batch bioreactor is that at the end of the experiment, when methane production reached a plateau, another addition of substrate was carried out. Finally, I also operate an anaerobic membrane bioreactor that, as you can see here, this is the biological reactor with the feed tank, and the bulk of the anaerobic reactor was circulated through a membrane, hollow fiber membrane, to keep the sludge inside the system and obtaining a high quality effluent, that is the permeate. Let's go now for the results. I will start with the first chapter. The title was that graphene oxide addition to anaerobic digestion of waste-activated sludge, the impact on methane production and the removal of emerging contaminants. This chapter was published in Environmental Pollution. To define the purpose of this study, I thought this study as an initial assessment, recreating the same conditions that happens in the wastewater treatment plant, using waste-activated sludge as substrate. That is the secondary sludge after the biological treatment. To assess the impact of graphene oxide, I prepared 3 different conditions: a control without graphene oxide, and 0.025 grams of graphene oxide per gram of volatile solids, and also 0.075 grams per gram of volatile solids. The main parameters were the methane production kinetics, the pharmaceutical compounds that were monitored at the beginning of the experiment and at the end in both liquid and solid phases, using SPE and labeled standard as a recoveries. Finally, I also monitored the microbial community changes with the quantification and the characterization of the 16S genes. Let's start with the methane. Here you can see a classical, this is how I represent the methane production. The symbols are the experimental methane production measured, and the lines represent the first order kinetic models that describe these kinetics. I use this model to quantitatively compare these methane production kinetics. As we can see here, adding graphene oxide, specifically 0.075 gram of graphene oxide per gram of volatile solids, reduces the specific methane production, the amount of methane that is produced per gram of substrate. The lower graphene oxide concentration test did not affect the methane production. But what it was also detected in both graphene oxide concentrations in comparison with the control is that the methane production rate was reduced by a 20% and a 40%. At this point of my thesis, at the beginning of my thesis, the main hypothesis to test was that this reduction in the specific methane production was due to the biological reduction of graphene oxide, that, as I mentioned, needs of substrate to reduce the graphene oxide structure. Regarding the removal of pharmaceuticals, you can see here that carbamazepine is one of the most studied compounds in wastewater treatment because it's very difficult to degrade, especially in biological processes. However, here what we saw is that in the control we achieve a removal around 20%, that is the typical value that is reported in the literature. But after the addition of 0.075 gram of graphene oxide per gram of volatile solids, the removal achieved was around the 60%. Moreover, there are other compounds that were enhanced by the addition of graphene oxide. Here you can see norfloxacin and ofloxacin, that is 2 antibiotics from fluoroquinolone family, and show to have a similar removal. This can be justified because the molecule is very similar in both cases. not all compounds, not all pharmaceuticals showed the same behavior. We also detect that some of the pharmaceuticals were only, the removal of some pharmaceuticals was only enhanced in the highest concentration of graphene oxide test, but the lower concentration of graphene oxide had the same removal as the control, as it's the case of azithromycin, citalopram, losartan, or valsartan. And finally, I want to end up with the bad news that not all pharmaceuticals were benefited. The removal of these pharmaceuticals was not benefited. It's the case of acetaminophen, clindamycin, and venlafaxine. Basically, what we saw is that 14 out of the 17 detected pharmaceuticals that in this measurement, in this analytics, 36 pharmaceuticals were monitored, but just 17 were quantified, were enhanced by the presence of graphene oxide, the highest concentration, 14 out of 17. But the presence of 0.025 gram of graphene oxide per gram of volatile solids just enhanced the removal of pharmaceuticals in 7 of them. Now, biological processes, when you change a condition, you should expect a change in the microbial community. The main thing that I detect, it's depicted here in the heat map. Red colors shows that a highest relative abundance, and blue colors lower relative abundance. I want to start highlighting that the addition of 0.075 gram of graphene oxide per gram of volatile solids reduces the relative abundance of microtricks. That it's it's widely spread, this microorganism causing huge problems in wastewater treatment plants. On the other hand, we also saw that some of these bacteria was benefited in the case of Rhombustia timonensis and Longilinea and Clandilinia. These bacteria are from Chloroflexia and Firmicutes phylum and are very well known as syntrophic bacteria that can exchange electrons using the direct interspecies electron transfer. Finally, this other plot represents the methanogens, the archaea community. What we saw is that in the inoculum, Methanosaeta concili was the main archaea present, and after the process with graphene oxide, decreases the relative abundance. The characteristics of this archaea is that it's acetoclastic, so it reduces acetic acid to produce methane and CO2. You can also see this group of here are all of them hydrogenotrophic archaea. And at the inoculum, the relative abundance was very low. But after the treatment was increased, all of them increased the relative abundance. For us, it was an indirect evidence of the direct interspecies electron transfer was occurring in our bioreactors after the addition of graphene oxide. Also, it's very important in environments when antibiotics are present, there is the proliferation of antibiotic-resistant genes. In this case, we decide to monitor 5 antibiotic-resistant genes conferring resistance to the main antibiotic families that are sulfonamides, macrolides, tetracyclines, fluoroquinolones, and carbapenems. And what we saw is that also the Intel one, that it's an indicator of gene transfer. Both of them, Intel 1 and Sul 1, did not show differences, and the relative abundance was pretty similar in the inoculum and also in the waste-activated sludge. And at the end, in our effluent, the relative abundance was pretty similar. On the other hand, we have ERM-B and TET-W, that the relative abundance at the end of the experiment, it was the same as in the inoculum. So we can conclude in this experiment that the addition of graphene oxide did not have an impact on the removal of antibiotic-resistant genes. So the main outcomes of this chapter is that the 0.025 gram of graphene oxide per gram of volatile solids concentration did not impact on the specific methane production, but decreased the methane production rate. Moreover, the highest graphene oxide concentration decreased both the specific methane production and the specific methane production rate, the kinetic rate. Regarding the pharmaceuticals, 14 out of the 17 monitored pharmaceuticals enhanced the removal, and syntrophic bacteria and hydrogenotrophic methanogens increased the relative abundance. Now it's time for the continuous biological treatment. This chapter has for title the impact of graphene oxide addition on pharmaceutical removal in an anaerobic membrane bioreactor, and it was published in Bioresource Technology. Here you can see a picture of my setup that includes the feed tank, the different pumps, the membrane, the hollow fibers membrane module. And here is more visual, you can identify the different parts. And basically, during the anaerobic membrane bioreactor operation, I was treating municipal wastewater collected from Girona's wastewater treatment plant. And the operation that lasts for 385 days was divided in 5 different stages that were the baseline, it's the startup of the reactor. We start spiking pharmaceuticals at a concentration of 0.02 micromolar, that it was an environmental relevance concentration. And then 3 different stages in the presence of graphene oxide, 0.005, 0.05, and then a weekly addition up to 0.1, dosing 0.005 grams of graphene oxide per gram of volatile solids. This is the operation. The dashed lines represent the different operational stages. And the main outcomes that can be shown in this plot is the efficiency removing organic matter, the chemical oxygen demand. And during the operational period, we achieve a removal over the 85%. Moreover, it's very important for the performance of an anaerobic membrane bioreactor to assess if this process is stable. To do so, we monitor the BFAs, but for the daily monitoring of the anaerobic membrane bioreactor, we use the ratio intermediate alkalinity, total alkalinity. That showed to be an indication of volatile fatty accumulation for values over 0.3. But, as you can see here, during the anaerobic membrane bioreactor operation, we kept always the reactor stability below 0.3, meaning that the addition of graphene oxide did not compromise the correct operation of an anaerobic membrane bioreactor. Regarding methane, we could not monitor the methane production during the anaerobic digestion process, but batch tests were carried out at the end of the experiment using cellulose, that is common and known in the protocols as carbon source, comparing fresh sludge from the wastewater treatment plant, the anaerobic sludge from the anaerobic membrane bioreactor after 385 days of operation, and also fresh sludge supplied with graphene oxide. In this case, what we used to model this data was the Gompertz model that includes a lag phase parameter, the maximum methane production rate, and the maximum methane production. The first thing that we detect is that the addition of the same amount of graphene oxide in the fresh sludge or after the adaptation process had different impact on the methane production rate. Anaerobic membrane bioreactor sludge had higher methane production rate compared to the graphene oxide, to the fresh sludge amended with graphene oxide. However, did not reach the same plots as the fresh anaerobic sludge. This can be justified because they were treating different sludge at different substrate. In the wastewater treatment plant, it treated waste-activated sludge, and in this case, was treating wastewater for more than a year. What we could also see is that the addition of waste of graphene oxide to fresh sludge increased the lag phase parameter. So the time between methane production starts and in the beginning of the experiment. This difference was not detected in the anaerobic MBR sludge. That means that the adaptation process has an impact on the methane production and reduces the negative impact of graphene oxide. And regarding the methane production efficiency, the specific methane production, again, the anaerobic membrane bioreactor sludge present highest methane production than the fresh sludge with graphene oxide, but did not reach the same amount of methane produced than in the control. This, for us, was an indication that it's necessary of the adaptation to graphene oxide. The mix of pharmaceuticals that we were spiking on the feed tank includes 7 pharmaceuticals that were sulfamethoxazole, trimethoprim, roxydromycin, and metronidazole. These are 4 antibiotics, and 3 pharmaceuticals that are triclosan, diclofenac, and naproxen. In this box plot, you can see the removal efficiency in the different stages. And the main result is that the 4 antibiotics showed highest removal efficiency when graphene oxide was dosed, but the pharmaceuticals, triclosan, diclofenac, and naproxen, were not favored by the addition of graphene oxide. And in triclosan, we saw that the removal efficiency was reduced by 10%. The main outcomes of this chapter is that the anaerobic membrane bioreactor achieved a removal efficiency over the 85% during all the operational period, and also we achieved the stability performance of the anaerobic membrane bioreactors. That is very important when you want to treat wastewater, because as I mentioned, the accumulation of BFAs can lead to the failure of the reactor. Another outcome is that the methanogenic activity was recovered after the adaptation, and the removal of 4 out of 7 pharmaceuticals was enhanced by the addition of graphene oxide. Let's start with the 3rd chapter. The title of this chapter is the distinctive effect of graphene oxide and reduced graphene oxide on methane production kinetics and pharmaceutical removal in anaerobic bioreactors. It was published in Bioresource Technology. The objective of this study, when we were defining the methodology, was to investigate the adaptation of graphene oxide in the same conditions at the same time. And we also monitored the pharmaceutical removal kinetics along the time in the presence of graphene oxide and hydrothermally reduced graphene oxide, comparing both materials with a control. To do so, again, I monitored the methane production kinetics. And in this case, we assessed also the expression of functional genes with metatranscriptomics analysis. The substrate that I was using in this case also was cellulose, because it's described to be a standard carbon source. And the graphene oxide concentration was 0.075, because in previous studies we saw that it was the concentration that has the better impact on the removal of pharmaceuticals. Here you can see also a heat map representing the gene expression after 24 hours in the control and after 72 hours in the control and also in the presence of graphene oxide. However, we could not detect major differences between conditions, but the main difference was in this classification of the KEGG database that includes folding, sorting, and degradation. And the main gene was associated with the twin arginine translocation pathway. This pathway was attributed to be in combination with peroxidase enzymes to be the responsible of an oxidative stress response. That I'm going to talk now about this. What's oxidative stress response? Is anaerobic bacteria, facultative anaerobic bacteria can live with the presence of oxygen, but there are others that can be killed, that can be completely inhibited by the presence of oxygen. So the oxidative stress is the disturbance in the protooxidants and antioxidants in favor of the oxidant species. The problem of this stress is that you can detect a reduction in the biogas production. Moreover, the oxidative stress response includes the expression, the synthesis of scavenging enzymes. These enzymes are responsible to degrade to consume this reactive oxygen species. Regarding the methane production kinetics, we carried out this experiment in 3 sequential fed batch bioreactors. When this is the plot of the first fed batch, this is the plot in the third batch. When the methane production reached the plateau, more substrate was added. The first thing that I can highlight here is the lag phase, that it was almost 3 days after the addition of graphene oxide in the first fed batch. As we know, graphene oxide needs to be reduced. And we know that it's occurring during the early contact of graphene oxide with anaerobic bacteria. In this case, what we detect also is that the methane production rate, the mu max, in this case of the Comper's model, was not enhanced, neither by the addition of hydrothermally reduced graphene oxide or graphene oxide in comparison with the control. However, after the adaptation, what we saw in the third batch is that the lag phase was the same for all 3 conditions, but also that the methane production rate was enhanced by a 37% in both the graphene oxide concentration, that in this case was already biologically reduced, and the hydrothermally reduced graphene oxide. That means that the biological reduction of graphene oxide is as effective as the hydrothermally reduction, without applying large amount of pressure or temperature. Now, the different pharmaceuticals that were spiked in this experiment were monitored for 72 hours, including the absorption on the surface of graphene oxide, because it has big absorption capability. What we saw in macrolides antibiotics, here you can see the 2 macrolides antibiotics are very big molecules with aromatical functional groups. The main outcome that we could see is that it was not absorbed in the sludge, but it was not degraded biologically in the control. And the main removal mechanism for roxydromycin was the absorption on hydrothermally reduced graphene oxide and graphene oxide. In the case of graphene oxide, reaching removals almost of the 90%. In the case of azithromycin, there was a biological reduction because there is difference in the control and in the blank without any conductive material. But we cannot disclose, we cannot guarantee that this removal is just because of biological reduction. After the adaptation, during the third batch, there were not differences between concentrations, meaning in supporting that the main removal mechanism was absorption at the beginning of the experiment during the first fed batch, and that direct interspecies electron transfer cannot enhance the removal of these 2 pharmaceuticals. Another family of antibiotics that was assessed was the sulfonamides that are widely spread as well. The main representative were sulfamethoxazole and sulfadiazine. In the main characteristics of these pharmaceuticals were that were not adsorbed in the graphene oxide structure, as you can see here in the case of sulfadiazine as well. And there is a huge difference between the kinetics. Sulfamethoxazole achieved 100% removal after 72 hours, but sulfadiazine was stable during the process. After the adaptation process, what we saw is more or less the same profile, indicating that the direct interspecies electron transfer cannot enhance the removal of sulfamethoxazole, but also it doesn't have an impact on sulfadiazine, that it's very persistent. The difference between these two pharmaceuticals can be justified because of the chemical structure. It was previously reported that there was a rearrangement in the iazole moiety in the sulfamethoxazole structure, so this difference in the removal kinetic was associated in this functional group. Finally, I also monitored two other pharmaceuticals that are represented, X-rate reagent ioxol and carbamazepine. And the main result of this point of the experiment was that both pharmaceuticals had more or less the same behavior, achieving a 60% removal at the beginning of the experiment, and were not absorbed on the surface of graphene oxide. After the adaptation process, what we saw is that carbamazepine was stable in both conditions, in the control and in the presence of graphene oxide. And during the first batch, it was not degraded neither. This is a very important result, and it's very interesting, and I'm going to go deep about this. Now it's time to disclose between metabolism and co-metabolism degradation. This profile of carbamazepine removal, the removal was highly enhanced by the addition of graphene oxide during the first 72 hours, was not detected after the biological reduction of graphene oxide. What means that maybe the removal of carbamazepine was linked with the addition of graphene oxide. A primary substrate can be degraded to a primary product and the bacteria can obtain energy. This is called metabolism. But in co-metabolism, the degradation of one substrate needs of another substrate. Without obtaining this secondary substrate did not provide energy to the bacteria. And the main conclusion of this result, the main hypothesis, was that the co-metabolic degradation was linked to the graphene oxide bioreduction. The main outcomes of this thesis, of this chapter, is that the biological reduction, bio-RGO, and hydrothermally reduced graphene oxide enhanced the methane production kinetics and had the same impact, showing comparable results. That the methane production after the adaptation rate was enhanced. The pharmaceutical removal was just enhanced in the first batch for both biological degradation and also absorption, but there were not difference were not detected in the third batch after the adaptation, meaning that direct interspecies electron transfer do not enhance the removal of pharmaceuticals. Now, it's time for the last chapter. That it's the title is the anaerobic biotransformation of carbamazepine catalyzed by graphene oxide, and this chapter is currently under review in Chemosphere. As I mentioned in the last chapter, in the last slide, is that the carbamazepine degradation showed to have a relation with graphene oxide, but we had to test this. To do so, we thought about a methodology with a sequential addition of graphene oxide at different points of the experiment. And also, we aimed for the elucidation of the major transformation products to ensure that it was a degradation process in carbamazepine removal. To do so, I used batch bioreactors using cellulose as well, and two different initial graphene oxide concentrations, 0.5 and 0.05 and 0.1, and adding graphene oxide at a dose of 0.05 at 76 and 167 hours. This slide is just for the results to clarify. Control will be depicted in black, and when graphene oxide was added at the middle of the experiment, the symbols will be hollow. And the same for the other graphene oxide concentrations. Regarding the methane production, what we saw, as I saw in previous chapters, is that the experimental data fits the Gompertz model, including a lag phase parameter. These dashed lines represent the graphene oxide additions, and this point in the control, we can see that the methane production kinetics, when graphene oxide was added in the control, reduces the methane production rate. This is because of the electron scavenging phenomenon, but it was interesting because the methane production was not completely inhibited. Just the methane production was just reduced. Regarding the other two concentrations, when graphene oxide was present at the beginning of the experiment, the addition of several pulses of graphene oxide resulted in longer lag phase in both initial concentrations, 0.05 and 0.1. Here you can see the other peak. It's important to highlight that here, the blue plot, it's 0.1 gram of graphene oxide per gram of volatile solid at the beginning of the experiment, and the black line had an accumulation graphene oxide dose of 0.1. So resulting in the same inhibition, the same specific methane production reduction, supporting our hypothesis of that the reduction of graphene oxide needs a certain amount of substrate to be reduced. Now it's time for the carbamazepine removal plots. In the previous experiment, the experiment lasts for 72 hours, and we obtained more or less the same results. It was stable in the control without the addition of graphene oxide, and the addition of graphene oxide enhanced the removal of pharmaceuticals. But what happened after that is that the graphene oxide, the carbamazepine concentration increased in those concentrations when it was removed, but it was stable for 240 hours in the control condition. This increase in carbamazepine concentration was related with a backwards reaction. But what happened when graphene oxide was dosed at 76 and 167 hours? In the control, we can clearly see the drop in the carbamazepine concentration, achieving a removal over the 60%, and the backwards reaction. To elucidate the major transformation products, this is the pathway that I thought that matched our results. In the control, we see an intermediate, this TP 250, but the concentration was very low. In the other, where the graphene oxide concentration was higher, the main transformation product was epoxy carbamazepine, 1011 epoxy carbamazepine, that it's known to be the major transformation product for this parent compound. And also that the highest concentration of transformation product was obtained in those conditions where graphene oxide was just dosed at the beginning of the experiment, meaning that a higher stress in these anaerobic bacteria can lead to further degradation because the transformation product's concentration was much lower. However, this is just an hypothesis. We cannot conclude that this happens because we cannot quantify, we cannot detect other transformation products. As I previously said, the reversible reaction, that's why we include a loop and also a backward reaction here. This reaction was reported in KEGG database to be catalyzed by cytochrome P450. So the main outcomes of this chapter was that the sequential addition of graphene oxide and the specific methane production was reduced, but the carbamazepine removal was linked to the graphene oxide addition, and mainly to the biological reduction of graphene oxide due to the scavenging enzymes responsible for graphene oxide bioreaction. However, it's a reversible reaction, but the overall removal reached over 95%, that it was never reported before. And finally, that the main transformation product was 1011 epoxy carbamazepine. Now, it's time to finish with the main conclusion of this thesis, is that the main conclusions are that the addition of graphene oxide causes an enrichment of syntrophic bacteria and hydrogenotrophic methanogen, that is evidence of direct interspecies electron transfer, that graphene oxide needs to be reduced to enhance the methane production rate, and biologically reduced graphene oxide is as effective as hydrothermally reduced graphene oxide regarding the methane production kinetic rates. Also, that the greatest enhancement concerning the pharmaceuticals removal was detected during the early contact between the anaerobic sludge and the fresh GO, and the removal of carbamazepine is enhanced by the co-metabolism linked with graphene oxide biological reduction. And finally, that the addition of graphene oxide to anaerobic membrane bioreactors to a continuous configuration did not compromise the correct performance of a biological reactor. It's time to acknowledge the funding to the Spanish Research Agency for the funding of the Antares project, that my thesis was within the scope of this Spanish project, and also to Generalitat de Catalunya and the European Social Fund for my scholarship, FI. Thank you very much for your attention. Thank you.
Do you want to take a seat?
No. I want to drink water.
We move now then to the second part of this PhD defense, and it's a little bit the questions and comments of the members. So, do you need a break or?
No, I'm okay.
Okay.
Surprisingly.
Very nice. So then we continue, and we will start then with Sergi, and you have around 20 minutes. We will see.
Ok. El primer de tot, hem pactat que faríem les paraules en català, perquè han vingut els seus pares, no?
Yes, my father.
Doncs patir potser 3 hores d'anglès no fa falta. I I'm gonna make some few words in català. Doncs, primer de tot, agrair a la Maite i a la Gerena que m'han convidat, que és el primer cop que estic a l'ICRA. Ha fet falta la teva tesi perquè jo vingui a l'ICRA, tot i que soc de Barcelona. I després també, Oriol, que ens va conèixer a Istanbul, no?
Sí.
Felicitar-te pel treball fet, no? O sigui, al final una tesi és un camí superllarg. Veig que no ha sigut fàcil. Cap tesi és fàcil.
Bueno,
hi ha moltes coses.
hi ha de tot.
Hi ha
hagut de tot durant aquest any.
Hi ha hagut de tot, no? Però sí que és veritat que, bueno, el volum de feina que hi ha és substancial, no? S'ha ficat de moda les tesis de 3 anys i no sé per què encara.
Això ho hauríem de...
A Catalunya fer unes tesis de 3 anys és un problema que hem d'arreglar. No sé si acabaràs fent la teva vida acadèmica professional d'aquest tema, però pensa que això és un gra de sorra dintre de la teva carrera. Segurament no treballis del que ha fet a la teva tesis, però al final el que t'emportaràs són les teves soft skills, no? O sigui, resiliència, capacitat de treballar en equip, les amistats que has fet durant el camí, la família, etc. Dit això, parlem en anglès, ¿vale?
Okay.
So, first of all, I want to acknowledge and I want to remark the amount of work you have done. The thesis is like you have done analytic digestion, like batch, continuous, feed batch, more continuous. And it's like, wow. We will talk about this later, okay? Then you have analyzed pharmaceutical products, which I've never done, but doesn't seem easy, in the liquid phase, in the solid phase, microbial ecology, antibiotic-resistant genes, transcript topics, okay, and finally, the last chapter, transformation products.
Yes.
Did you sleep?
At the beginning of my thesis, especially the anaerobic MBR, was a nightmare.
The what?
Was a nightmare. I was dreaming. I was dreaming that the anaerobic membrane bioreactor decide to overflow or to get empty. And actually it was happening at the beginning. So actually the baseline was longer, but I cut it because there was a lot of, I failed many times, but then I decided to change the control strategy and it was working much better.
Okay. Now that you mentioned the MBR now, there's going to be more discussion. Why could you not measure the methane production? It happens. We also had the same problem in Barcelona.
First reason that I thought it was because I was using municipal wastewater, so the amount of COD that the organic loading rate was very low in comparison with other anaerobic digestion reactors. But the main problem that I had was that it was very difficult to keep the volume constant. Very low variations in the volume can affect on the methane production kinetics because I was trying to measure it with a flow meter. But if a little bit of influent was going inside compared with the permeate that I was achieving, then I was overestimating the methane production. Then I had underestimation because my reactor was getting empty. And it's very tricky to work with the membrane because it's getting clogged. And then at the beginning, it permeates a certain volume, but after a certain amount of time, then the permeation efficiency is reduced. Yeah. So more liquid is accumulated inside your reactor and you are overestimating. Yeah. So that's why I decided to perform batch test at the end of the operation and compare it with fresh sludge.
Yeah, that's fair. We all have troubles measuring gas production in membrane bioreactors. So that maybe lies one of the critiques that I would have, and also the external examiners have raised that. The materials and methods could be better explained. It was hard to sometimes understand, like I don't recall reading the HRT or like organic loading rate of the SRT or sludge retention time of the bioreactor that I was looking for. It is like, okay, I miss it. And it happens like in few of them. I was missing information, like the last one, which I think is my favorite graph, the one that you show with the feed batch pharmaceutical products removal. Yeah. I wasn't sure how you did the biotic and the abiotic experiments. I was looking for them because I thought it was very interesting, but I will come back to that. So it was sometimes I miss some information that I would have loved to find. Okay. Maybe also like it comes to my mind. It's a ICRA thing, or why are not the papers in the thesis?
Because it's not an ICRA thing. It's UDG issue. Okay. Because you can use there are two different ways: the Scandinavian way to publish a thesis, that you just put your papers there, or using a monographic thesis. Yeah. And we can do also a mix. When you can write a monographic, that it's, in my opinion, it's also easier to read this thesis in comparison with just putting a PDF here that the letter size is super small.
No, I was just expecting them at the very end of the document.
But yeah, but then the thesis increases. Yeah.
Yeah, don't worry, they are online.
Actually, it's the same. Like this text is from the manuscript, so it's the same text. Okay. Results.
Okay. Maybe because I was curious about the materials and methods. So. Yeah. One thing that it's sometimes we miss it and we don't pay attention, but I think it's very important. The context. So I was a bit confused about the context of your thesis. Let me explain. So you did a lot of substrate, a lot of different experiments, but which is the final goal? So use graphene oxide, pharmaceutical product removal, gravity. It was my back, don't worry. So we are shifting the complex from wastewater treatment to resource recovery, and then biogas is one of the key products in water resource recovery facilities, they call it now, biorefineries, whatever. But then, you are at ICRA, so water is also super important. Water quality is also very important. So and then you use MBR, but the BMPs are done with WAS, waste activated sludge, and then you do other with cellulose. So I was not certain about the context. What was your application focus? Or you know what I mean?
Yeah. The main objective of my thesis was to assess the impact of graphene oxide in municipal wastewater treatment. However, when I decided for the first results chapter, I used waste-activated sludge to reduce one variable. So I was using the same substrate that is used in wastewater treatment plant, the same inoculum from the wastewater treatment plant. So the inoculum was acclimated with to degrade this substrate. And also because this waste-activated sludge contains pharmaceuticals compounds. So the idea was at the first chapter, it's the first assessment of what happened when you add graphene oxide to an anaerobic membrane bioreactor, an anaerobic digestion reactor. Then in parallel, I was operating the municipal wastewater treatment plant, that it was an anaerobic MBR. But after this, after I finished the study from the anaerobic membrane, anaerobic MBR, to have repeatability, to repeat these experiments and to ensure that I don't have pharmaceuticals coming from the substrate, different organic loading rates between replicates in the first fed batch or in the second fed batch. I found the IWA protocol that describes cellulose as carbon source for biochemical methane production test. And the other advantage, it was that I didn't have pharmaceuticals from the substrate, just the ones that were already in the inoculum, but then I can have the more stable conditions and just one kind of biomass. That's why at the end, I use cellulose. Also, if the experiment lasts 12 days, I know that after 12 days, I can add the same amount of substrate with the same characteristics, and the plots would be the comparable.
Yeah. So it makes sense just to use some synthetic substrates to reduce uncertainty. So focus on what you were focusing.
Yes.
I mean, I'm sorry for the next PhD student, because I think that the way to go is to go harder in the anaerobic membrane bioreactor, because it makes a lot of sense, because there is a lot of research adding carbon particles, not just to support biomass, also for membrane scoring, to clean the to clean the membrane.
Yeah.
So there are a lot of technologies, and you have a product that is conductive, it's apparently better. No, a lot of research with activated carbon, for instance, biochar, such. So, what about the conductive properties of reduced graphene oxide and graphene oxide? Because I was looking for a table in the introduction.
Yeah.
And this goes with a second one, with the lasting effect. You know, like when you add, you had an effect, but the lasting effect, because in the batch feed reactors are a bit confusing, the lasting effect. What's your opinion? So,
What
two
was
questions.
the first question?
It's about the conductive properties of graphene oxide and reduced graphene oxide.
Yeah, it was I think it was in the supplementary material of one of the papers, and then in the thesis I forgot to include it.
Okay.
But it's very difficult to quantify the quantification of a nanoparticle. It's very difficult to be quantified. But what we did is with other PhD student that was working more or less on the same, the characterization using Raman spectroscopy, how oxygen functional groups are reduced, and 2D structure was recovered after hydrothermal process, and also there was he detect the biological reduction by Raman spectroscopy. But about the conductivity, I did not characterize.
Have you read about it?
Yes, I read about it, but it's not it doesn't have the same superconductivity properties than graphene because it has defects. There are some oxygen functional groups, but also there are holes in the hexagonal net. So it's not that good as graphene, but it had good conductivity properties. I don't remember the number now.
That's okay. So it's cheaper, that you mentioned, no?
Yeah, it's cheaper because it's produced to chemical oxidation from graphite. And then you can biologically reduce. If not graphene, to produce graphene, pure graphene, it needs to be produced through exfoliation. And to produce one layer thick of a nanomaterial, it's very tricky and also very expensive.
Well, regarding, I'm going to make some random questions. Chapter 1, you use a bioprocess control system for the BMPs? I think that you mentioned that in the chapter 1.
No, I was using bioprocess control system in the 3rd and 4th chapter. At the beginning of my thesis, I was carrying out this BMP test manually, measuring the overpressure in the head space through a bromobutyl septum.
Yeah, okay.
And also quantifying the methane concentration in the head space using a methane proof.
And at the end, you use the...
At the end, we decided to buy this fantastic equipment.
This fantastic equipment. But...
Very expensive as well.
Very expensive as well. So in the feed batch you did it with this experiment?
The fed batch, I was using the bioprocessing control.
And the thing is like, so I was, yes, I'm wondering because I was expecting more data frequency, you know, bioprocess controls give you every 3 milliliters or whatsoever.
Yeah.
I was expecting a lot of data points, which is very good to feed the kinetics, but then you have like...
This is the daily production.
Okay.
Because if not, as you say, there are a lot of points that in my opinion are not, experimental data can be represented with points, but what describes the results of your experiment is to describe your experimental data with a mathematical model.
Yeah, that's fine. But I was...
Yeah, that's why I reduced also because I was using Excel, so the amount of data I was treating was low, but it was as much representative. The general result, it was the same.
I'm sure. I was just curious. No, sometimes the model, no, the more data you put, the better it fits. Sometimes, but I think that the trend is the trend.
Yeah, the more data you put, also the fitting is better.
Exactly. Let me check. So you did a lot of microbial ecology studies, and in the defense, you didn't talk or didn't mention much about which I think is the most relevant one, which is the one under continuous experiments, which is the one from the anaerobic membrane bioreactor.
Yes.
You observed, did you recall, you haven't mentioned it here today, isn't it?
No, I haven't.
Did you recall the same pattern, also like more syntrophic acetate oxidation, more hydrogenotrophic methanogens?
It was tricky because the operation of this reactor was over 385 days, also using real substrate that contains bacteria and archaea probably. And the results were not that clear. We saw a tendency in the proliferation of syntrophic bacteria and also hydrogenotrophic methanogens, but it was more visual in the first chapter than in the 3rd chapter, so that's why I decided to skip because it was just tendency. In percentage, the changes were not that important at phylum level. Then some species showed tendencies, but it's just this. I cannot ensure that this was because graphene oxide, also because of the many other parameters, because the substrate was changing every day.
And how many times did you add graphene oxide? Every day?
Not every day, every week.
Every week.
So at the beginning of the third stage, I had I don't remember the volume of graphene oxide, but to reach overall dose of 0.005, then another addition to increase an order of magnitude the graphene oxide concentration, and the last stage, the weekly addition of graphene oxide.
Okay.
So,
But no matter what, you have 85% removal of COD, and a stable reactor, a functional reactor that remove more pharmaceuticals, 4 out of 7, isn't it?
there was a tendency in the removal of pharmaceuticals.
Which is overall is good.
Yeah.
Of course, maybe you have less methane, but in an MBR.
But I thought, yeah, of course, as it can be shown in the last chapter, it clearly affects the addition of graphene oxide.
Yep.
But in stages 3 and 4, methane production would just be compromised at the beginning of the experiment. At the beginning of the stage, during the first 2 days. The main implications that I also would like to try is that if the process is faster, you can increase the organic loading rate.
Yeah.
But I could not test it because of the membrane.
Yeah.
At that point, I was having, I started having some problems.
Yeah.
And I was really interested in testing the organic loading rate, but at the end, I tried to keep the system, the setup, as stable as possible.
Yeah. This reactor was mesophilic or?
Mesophilic at 37 degrees.
I think that the research will be more interesting if it goes syncrophilic. It's going to be slower, and maybe the addition of the additive can be more remarkable. Also, it applies better to wastewater treatment. No, it's hard to.
Completely agree.
I think that this benefit that you mentioned can be very interesting. Like, okay, better kinetics. Like at mesophilic, everything works. But when you go down to 20 degrees or whatsoever, these tiny effects can have a very positive impact.
These are some parameters that we were thinking to assess when we start operating the anaerobic membrane bioreactor. But as I said, after 1 year of operation, the setup needs a lot of love. We had to take care of it.
It needs care.
And to change these kind of parameters needs to extend also the operational period, and it would be very tricky. So that's why I decided to stop the anaerobic membrane bioreactor and go for the fed batch bioreactors to go more deeply into the mechanisms.
I mean, we sort of somehow solve these problems at our research lab, but basically the technician needs to be there when it's feeding and when it's permeating. And we have a PhD and a technician in this project. So
I was the PhD and the technician.
But that's I mean, and even though it is
Yeah.
it is tricky.
That's why after a year I decided, okay, that's the end. I don't want to invest my life in this, because I was doing this actually.
Don't tell to my technician that wants to be a PhD student on the topic. But then in this BMP that you mentioned, somehow these ones.
Yes.
I don't remember the slide here. These ones. There is somehow of an adaptation to the graphene oxide addition.
Yeah.
And that's interesting. I think that, and it comes back to my mind the lasting effect of graphene oxide. That's super hard to test. I think that, but that's a topic.
That's
actually
this,
a topic for another day.
This was the plot that encouraged me to go for the fed batch reactors.
Okay.
Especially the shortest lag phase, not the methane production or the methane production rate, but the lag phase. That means that it was not affecting. Graphene oxide was not affecting when it's biologically reduced and adapted. So then I changed the configuration and I test this.
Yeah. Still, like as an engineer, adding a compound that steals electrons from methane, it's a bit challenging, but it's only the first after the dose. You sacrifice something to win something. That's what it needs to be.
That's what I thought after the first chapter.
It's a win-win-win-win. It doesn't exist. It's not real. And water rate. Sorry. Last minute. Last question. Last question. Damn it. No, last question. Otherwise. Sorry, I'm too curious. Okay, let's finish. So, felicitats. Jo encara me'n recordo quan feia el doctorat i passava 1.000 milions d'hores al laboratori i no feia tantes coses com tu. Llavors, felicitats, que ho has fet molt bé, m'ha agradat. El document, felicitar-te perquè és molt fàcil de llegir. Fa tant materials i mètodes, so I miss a material method, but the document is easy to read. So I will suggest this document to my PhD students, so I have to read less. And I make read less to examiners and such. So congratulations.
Thank you very much, Sergio.
So next would be Dr. Anna, please.
Yes, thank you. And well, first of all, thank you very much for your nice work. It was really a great pleasure and with great interest that I studied it already earlier in October. Thank you also for Maite and Helena for inviting me. So I have to say that I really learned quite a lot from this work. And I also have to agree that it's kind of an impressive set of different experimental studies, the analytical methods that you have discovered, or well, maybe not totally discovered during your work, but really kind of carried them out in great details in the work is amazing, I would say. So I think that you have really kind of constructed a fairly firm ground for your next steps as a researcher. So that's really excellent. I also think that it was kind of very clear and in a way logical thesis to read. But I also do agree that there were some things, some details about the methods, probably because there were so huge amount of different things that you did that it was it took maybe 3 or 4 readings before it was
and
kind
5.
of easy to understand everything. I would maybe kind of like to challenge you anyway on a couple of points. So this was actually some things that I kind of discovered during your presentation, which was also I think that you kind of improved the clarity in your presentation today, which was also very good. I think that it also contained some additional information that you didn't include here. It was helpful and interesting, but I mean, well, I guess that they could have been here, but we can also understand the kind of progress of the work. So, well, one of the goals was to kind of, let's say, prove that your process can work as a stable process with real wastewater and so on, and that was one of your conclusions in the presentation and here as well. And for that, actually, I would like to kind of know a little bit more details about how the continuous flow process was actually operated. So now you mentioned that you collected fresh wastewater once a day, and then it was pumped to the reactor. But was the pump? Oh, so how was that actually done?
It was I was collecting once per week or every 2 weeks. I was going to the wastewater treatment plant to collect fresh water, keep it in the fridge, and then use it put it in the feed tank, and spiking the pharmaceuticals to ensure that I was detecting these pharmaceuticals.
Yes.
The operation of this reactor it was 3 stages. The control program includes 3 stages. That was the feed, that it lasts for a minute. The reactor was fed during a minute. Then it was a mixing stage that it lasts 2 minutes to keep homogeneous conditions. And then the permeate pump, and also there was an electrovalve in the permeate line, was extracting the permeate. So every 5 minutes, I had a cycle. So I would call it this a continuous reactor also, although it have different stages, so it can be also called SBR, but in my opinion, the volume was not changing that much. The media was almost the same because the volume exchange was not big, did not have a big impact because the sludge, the hydraulic retention time was 1 day.
Okay.
So yeah, well, the operational conditions was like this. Also, to keep the membrane, the permeability of the membrane, I was recirculating all the time the bulk of our anaerobic reactor, but also the head space to increase the turbulence,
Yes.
and then to avoid the microorganisms to grow on the surface of the membrane. That it's known to be one of the main problems working with anaerobic membrane bioreactors.
Okay.
And then I was doing also some cleanings during the operational period when the transmembrane pressure was below a certain point. So that's not detailed. It's detailed in the paper, but I don't know if I include it in the thesis.
Yes, yeah. But now it seems that you really kind of, or the operation was very close to, let's say, real full-scale operation. So you were kind of able to show what kind of issues might cause challenges. I think that you in the conclusions or in a further research suggestions, you also talk about these potential challenges. But my point maybe would be that do you think that you were able to, with kind of collecting the sample every second week of wastewater, were you kind of able to mimic the normal variation in water quality and in kind of water quantities also that we have to deal with in wastewater treatment plants?
Definitely.
Because now it was kind of a stable flow that you had into the process.
Definitely, this is the quality of the effluent changes during the day, but this parameter was not included because I was always collecting my wastewater at between 10, 11 a.m. So expecting to have more or less the same matrix.
Yes.
Just to avoid operational problems. Because one of the days, in wastewater treatment plant, there was the centrifuge from the anaerobic digestion that treats the sludge were on, and all reject water was going to the influent of the plant. So there was one parameter that was like, okay, I don't want to deal with it. I will collect water always the same as I can to avoid this variation during the day.
Yes. Yeah.
And the operation of this anaerobic MR did not include differences during the day. It was always the same in weekends, at night, always the same.
Yes, yeah, and I think that was a very good choice for the research to kind of avoid those problems.
Yeah.
But then I guess that it's fair to say also that before this process kind of can be really, let's say, stated as a stable process, these kind of tests would still need to be done to see how it actually reacts.
I completely agree.
I mean, there can be some equalization basins that can be used, but that's, let's say, typically the wastewater anyway needs to go somewhere, even if it's not of the quality that we would like to have in the process at that point. So yeah, that's our common challenge, I think, in the field. Then the second question that I would have for it is for the pharmaceuticals. Yeah. So I think that you really showed interesting results and a nice, also very kind of deep analysis of kind of what could explain the results and so on. But in the first place, when you selected the pharmaceuticals that you wanted to study, how did you select them? Did you already know beforehand what were the mechanisms that you would kind of like to show or study? How kind of systematically you actually chose the
first in the first study, I used a method that was already developed here in ICRA by Mary Gross, who's a researcher here in ICRA, that the method includes 36 pharmaceuticals that are widely spread in environmental samples and also in black water. And this method includes the SPE extraction and the solid phase extraction from the pellet or the solid fraction of the sludge, using recoveries. So in the first study, I did not choose the pharmaceuticals. It was part of the method that was already developed, and it showed very good results in other samples. Then in the anaerobic MBR, to be honest, we were monitoring more pharmaceuticals than this. But at that point, I was seeing very difficult data to be interpreted. And then I discard specifically tetracycline. But yeah, the molecular structure of these pharmaceuticals that I showed contains oxygenal functional groups mainly. And we thought at that point that were interesting because may be reduced during anaerobic processes. So the criteria for this second chapter was the chemical structure. And for the third chapter, I included 2 representative or 3 from fluoroquinolones, tetracyclines, sulfonamides, and macrolides antibiotics, 2 of each, and 3 fluoroquinolones, and 2 pharmaceuticals that were ioxol and carbamazepine. I choose these pharmaceuticals because I showed potential. I saw potential in the first study. So I decided to include carbamazepine in this study. And it was a good choice because it was the reason why I could also go deeper on these transformation products in the last chapter using carbamazepine.
Yes, yeah. So I'm kind of happy to hear that it seems that you really kind of you really did a lot of thinking, and you were not just randomly picking some compounds. But then it, of course, because it was, when I was reading it, it was a little bit confusing to see that, okay, you first had 36, and then you had something else in the 2 other studies. So it would have been kind of really good to write down and explain in the thesis and maybe also in the publications this kind of systematic approach, because that is, I mean, you clearly did a really great job on picking and thinking about the compounds, but it would have been really nice to see it here as well. Yeah. But I guess that's too late now.
It's too late, and
for the next
I think that I have 6 versions of Mathilians and methods section. And at the end, I was asking my supervisors that, can you help me with this? I cannot decide what is the best way to present my materials and methods. That's why I describe a general materials and methods in the chapter 2, section 2. And then in each results chapter to give an overview. I was doing the same during the presentation to like, okay, there is a lot of materials and methods. I was using a lot of things. But in this chapter, what is important is this.
Yes, yeah, and overall that approach of having first the general introduction of the whole thing worked really well. So I think that it was a good choice. But maybe there was just too much to tell for one thesis. Then could maybe, ask one or have maybe more of a comment, or also ask your opinion on so you did quite a lot of microbial population studies, and so now I'm looking at the figure 4.3 where you have I think the first batch results with the control and then the graphene oxide addition. So here I guess that you had 2 reactors, so kind of 2 batch reactors of in each condition. So you've had the initial.
It was the initial mix that it contained inoculum and waste activated sludge. Yes. And then It was collected from the control, I guess. The initial concentration was the same in all reactors. And then at the end of the experiment, I pulled all the triplicate of the control and also the highest concentration of graphene oxide, skipping the lowest graphene oxide concentration because I was not expecting differences or huge differences in the lowest graphene oxide concentration. And then we quantified the 16S reactor, the 16S genes in the two final concentrations.
Yes. Yeah. Yeah, I was kind of just, so I think that here in a way the differences are so, let's say, logical and so clear between the control and the graphene oxide reactor that, I mean, kind of probably, or it kind of seems perfectly okay to draw your conclusions, but are you really kind of absolutely sure that if you, let's say, had two parallel reactors in the same conditions each time, kind of how big differences you would have seen in the microbial population in, let's say, kind of two control reactors and then two...
Continuous reactor, you mean, or batch?
Or the batch. I mean,
these actually were in parallel.
Yes.
At the same time, with the same inoculum, with the same waste-activated sludge, and the difference were clearly detected. Between replicates in the same concentration, in the same condition, you can expect differences. Because in biological processes, it can happen everything.
Yes.
It's something that I learned, that also there are some studies conducting the same experiment in different geographically spots, in different universities, and the results were not the same. Using the same sludge, the same substrates, and the results were not comparable because of geographical differences. If you set up two reactors at the same time, running in parallel, you can expect differences in the microbial community, for sure.
Yes, yes, exactly. So this was exactly kind of what I wanted to, or
Yeah,
was
yeah.
hoping to hear. So it is clear that I mean, the microbes will be impacted by kind of hundreds of things that we don't understand and know. So they will live their lives, and in a way that's why it's tricky to draw conclusions. I think that in this case it's really kind of very clear and okay. But I also think that your decision of not using the continuous flow reactor results or try to draw conclusions from there was probably the correct decision, because I can really well understand that it's really tricky to see
the
anything for sure.
The slide was included in the first version of the presentation, but I was struggling with highlighting the results. So at the end, we decided just, okay, let's delete it. And we skip this part, and if we need to discuss it later, we will discuss it, but without presenting it.
Yes. Yeah. And what would be then your solution to, I mean, if in the future you would need to show in a continuous flow process the impact on microbial population, what would be your solution to kind of see something with those results, considering that you don't kind of know this natural variation when the influent qualities is changing and so on?
The first thing that I would do is to monitor the microbial community in the influent, to know which species are going inside our system, to assess if there is a consortia replacement, because maybe there is suddenly we have a peak that methanotrix can be the most common, with the highest relative abundance. But if we know that it comes from our substrate, we cannot attribute it to our operational conditions. Another option is to use the real substrate, but disinfected. It's the only way, I guess, that you can assess the microbial community changes in a long-term operation without taking into account the changes attributed to the influent.
Yes, yeah, I think that they are kind of good suggestions. And then, well, maybe just following also the process over.
Yeah, and more
years,
the
4
sampling
years, so then
and the sampling frequency. I think that it should be also more maybe weekly or every twice per week. You need more sample to do a microbial ecology study. This was just to present an overview to give us an idea at the end what's happening with the microbial community.
Yes, yes. Yeah. Maybe have one final question. Then let's see. Maybe so. If we go to the kind of methane production and the so I'm on the page 122.
Yes.
So what? I mean, also in the presentation. You were talking or showed this kind of dead time caused or kind of the adaptation phase that is needed also depending on the substrate that we have. Well, actually, one question that I now kind of saw here noticed during the presentation, it's the figure 6.2. It kind of seems that actually when you repeated the test, then it's true that kind of all three have the same delay, but
we
it's
have
actually
delay.
also kind of the control, or there was kind of less delay in the graphene oxide addition, but more delay at the same time for the control.
Yes.
Is this just
This
so,
can
so
be.
what could explain that actually?
That can be explained because the sludge, when I was collecting the sludge from the wastewater treatment plant, I kept this sludge in an incubator at 37 degrees for 10 days to degrade the organic matter present there that comes from the anaerobic digestion reactor. And at the end, this container was connected to a bubbler to see if methane was being produced. And I said, and also the protocols detail that you have to wait for 10 days. Now I don't remember exactly how many days I wait to start this reactor, but maybe I wait just 8 days. And that's why in the first fed batch, the methane production started a little bit before. But also, it was not that significant. It was more or less, we can clearly see here, where is the pointer? Here is not that significant. It was 2 days, more or less. But this amount of methane produced could be attributed to the substrate that comes from the inoculum. And this lag, specifically this lag phase, that it was not detected in the first study, it's because of the substrate, because in this case, this is cellulose, that is not soluble substrate, so we have mass transfer limitation. That's why we have a little bit of lag phase before. But again, it's a substrate that is described in protocols, and it's very easy to work with it. Also to replicate results. But yeah, this lag phase is something. It was a very interesting parameter to assess, actually.
Yes, yeah, that was all. I would have had a couple of more questions about the lag phase and the dead time, how to kind of try to implement that in full scale. But I guess that now I will leave some time for Marta also for her questions. Maybe she can continue on this interesting topic. But yes, thank you for this discussion. We really had excellent answers here, and you really showed that you have a incredibly deep understanding of the topic. So congratulations.
Thank you very much.
Okay, thank you. So I also want to have some time for me to ask some questions. That's just for. So first of all, thank you very much, Maite, Jelena. It's also my first time here as a member of a PhD defense, and it's my first time as president. So, but I still have questions, don't worry, to ask, because you are the last and so on. So I agree with my colleagues that the thesis is an incredible amount of work, touching different topics, because you touch technical things, operating different types of reactors. You touch emerging contaminants, not only pharmaceuticals, with target, non-target analysis, transformation products that you never is there. Antibiotic-resistant genes is a step further on that. And then even microbial community analysis. So from DNA, even RNA, metatranscriptomics. So I think, it fulfills the 3 already published papers plus the 4th ongoing. We don't have much to say. There are already, everything has been already said before. But I still have some questions.
Okay.
So I will start with some small questions, maybe to go, and then some helicopter questions, as I call them. So when you try to, when I think on carbon-based materials, put in an anaerobic digester, I say, okay, sorption will be there as a main mechanism. But then you try to characterize sorption, if I understood correctly, by the log D, right? With the log D of the contaminants. And you say that that I associate it with the low KOW, the octanol
But
water.
the log D was used for the interaction with the sludge.
Interaction with the sludge, sorption on the sludge.
Yes.
But so log D is equivalent to the log KOW, because I could not find in the acronyms the log D. So that, I don't know. That was because my question was mainly sorption has the 2 parts, right? You say the lipophilic part when you and the electrostatic part, no, the absorption and the adsorption. So my question was just why you choose the log D and not the low KD that includes both mechanisms to characterize the sorption onto the sludge. If there is
this question, you are asking me something that I was doing 3 years ago. So I can remember, I think that I remember that the log D is not the lead. The other one, the KWO, is the partition coefficient between octanol and water. So this is the lipophilic affinity. I was using log D with to assess the charge.
Okay.
The electrostatic, as far as I know, is or I can remember now, is the electrostatic interaction between materials. And actually, this is a result that it's detailed in a supplementary table that the different pharmaceuticals had different removal, different concentration. Some of them were more concentrated in the sludge. Some others more present in, for example, carbamazepine is the clear example that it's stable and it's in the liquid fraction all the time and it's not absorbed in the sludge.
Yeah, okay. Yeah, but it was due to that. Yes, I continue with some small questions. When you think on an anaerobic MBR to treat wastewater, what would you place this technology in the wastewater treatment plant layout?
Definitely I would, in an anaerobic membrane bioreactor, I will place it at the beginning.
At the beginning?
Yeah.
After, so I will specify a little bit, but before or after primary clarification? Where do you place it?
Good point. Primary clarification, the primary clarifier remove a lot of particulate organic matter. It also depends on which kind of membrane you have.
Yeah, that's true.
If you have a module that I had, like I had, if you don't remove the organic, the particulate matter, then for sure you will have problems inside your cartridge. If you have a flat membrane or submerged membrane, you can skip the clarifier and the mainstream, place an anaerobic MBR in the mainstream without the primary clarifier, but then you will have probably problems with the volatile solids and total solids accumulation inside your reactor.
Yeah, because if I think on also methane production, right?
Yeah.
So you are already discarding some potential organic matter in the primary clarifier to go there. So my first view was to, let's say, replace primary clarifier and also for the anaerobic membrane. And also depends when where you place the membrane, right? So you can put it as an external chamber.
External chamber, submerged,
Exactly.
hollow fibers,
Because,
flat membranes. But also I think that it's very important to consider the quality or the characteristics of your influent.
yeah, yeah,
Because as I said, if you have a lot of particulate matter, a lot of paper, then you will have definitely that the biodegradability is poor biodegradable organic matter or even
yeah,
mineral fraction.
Yeah.
It contains a lot of sand and clay. You will have solids accumulation inside your reactor, and at the end, that volume
yeah.
in your
yes,
reactor.
You have to put. Yeah, I was thinking mainly after pretreatment, right? So just
Yes.
I will say on that. Yeah. Just one question. In terms of micropollutant, well, emerging contaminants removal, just if my questions are, you follow the same strategy as in chapter 3? I mean, measure liquid and solid phase in all the chapters when you state removal efficiency, or in chapter 5, 6, and 7 is only liquid phase? It's just because of my questions can go in two directions depending on that. Because in chapter 4, I think in the first chapter,
The
it's
first chapter,
clearly
it's
clarified
both.
that is
Yes.
liquid and solid, so the removal is biotransformation because you count for the other mechanisms.
In anaerobic MBR, I'm talking all the time about removal efficiency without clarifying the
Okay, yeah.
mechanism, because it's just
Liquid
in the influent and in the effluent. So it's liquid phase without the accumulation in the sludge. And in the other ones, chapter 6, it's just in the liquid phase, but with the absorption test
Yeah, with this.
into a graphene-based material.
Perfect. Okay.
And also in the controlling, the absorption was monitored also in the cellulose, because it may have an interaction.
Yeah. Okay. Because probably you notice also with naproxen and diclofenac, in the first chapter you have removal, no? And in the second chapter, in the anaerobic MBR, you don't have any removal. I have some experience with those two contaminants also in anaerobic digestion.
I know.
So, do you have an explanation why I don't remember the number of the chapters, the chapter 4, I think you have removal of these two anti-inflammatories, and in chapter 5, in the anaerobic MBR, you don't have. And it's clear, huh? So, I mean, it's not that you say, maybe some doubts for
Actually,
these two contaminants.
These two contaminants, I would like to follow the same strategy than in the last result chapter and assess if it's a co-metabolic degradation. Because it was clearly, specifically diclofenac,
Yes.
the removal was 0% in the control and in the lowest concentration, and then super high. So I cannot disclose if it's the removal, it's adsorption or not.
Yeah, because when you see the removal is biotransformation, because it's in the chapter in which you measure both phases.
I guess, I assume that it's biotransformation because I'm quantifying in both matrices, solid and liquid, and using labeled standards. So I would say that it's biotransformation, but I would like to do more experiments to completely affirm and conclude that it's biotransformation.
I was thinking another possibility explanation also for that. Do you think the substrate can have?
Yes.
Because that's a real clear difference between the two chapters. In one chapter you have activated sludge, which I would expect more enzymatic
Variety.
involved in their degradation.
The same happened with carbamazepine. 20% in the first chapter and then 0% in the other one. It's co-metabolic degradation, in my opinion, but I don't have evidence to...
metabolic degradation when graphene oxide is present.
In the case of carbamazepine, because...
No, and diclofenac and naproxen also.
Yeah.
In both.
That's why I said that I want to, but I cannot say it because I haven't done
it's very interesting, those three.
I haven't done the experiment, so I cannot say it. It's in my list, it's in my to-do list.
And is co-metabolic not related to methane production? Because methane production is higher in the control, not the...
Yes.
So those three are really to pay attention on that. I find it very interesting, those three. something more. In general, do you think when you compare graphene oxide and reduced graphene oxide in that chapter, the sorption capacity of both materials, do you expect it similar? Do you expect it different?
No, it's different.
Why is different?
Mainly because the difference between hydrothermally reduced graphene oxide and graphene oxide is the oxygenal functional groups.
So reduce is better, sorption
reduce,
has...
well, depending on which kind of interaction you want to assess. Because the hexagonal structure of reduced graphene oxide promotes the PP interaction with other aromatic structures, but oxygenal functional groups can have a weak interaction as electron, hydrogen bonds, or interaction with other functional groups.
Okay, so
that's why specifically macrolides, that they have a lot of oxygenal functional groups in the structure, the removal was attributed to, that it's the next slide actually. This. The removal was clearly enhanced by the interaction with graphene oxide, and reduced graphene oxide, the removal was 25%.
In general, I think that the reduce is better except trimethoprim, if I remember that well. It was in general much better than the graphene oxide. But it's nice, this graph, because here you can, if you compare, let's say, the abiotic against the biotic, but sorption on the inoculum is
it's another thing.
It's not accounted. I mean, it's not accounted in the hollow dots. It's accounted also. So the hollow dots is clear. Is abiotic, but the bold dots is sorption on biomass plus biotransformation, right?
I think that I didn't mention biodegradation in this slide.
No. It's just
because I was very aware of this. Like, it's related with the sludge, but
Exactly.
I don't know the mechanism.
The mechanism, yeah.
And the problem we were discussing also during the methodology, when we were defining the methodology of these studies, to include inactivated sludge.
Yeah.
But then it's very difficult to keep the same structure of the cells, because using an autoclave, you are breaking the cells, you are releasing a lot of biological material into the bulk. You can have transformation.
Yeah, I agree.
That's why I just skipped this part, and I decided to go for blanks to test adsorption, and then biological mediated mechanism, without disclosing with degradation or not.
Yeah, we have experience with the thermal inhibitor, and it's the same. So they don't have the same structure. The KID values are completely different. Anyway, also taking this graph, you see the abiotic, let's say, after 48 hours, no, more or less. It's quite slower kinetics to be an abiotic biotransformation, don't you think? I mean, for me, for abiotic transformation, physicochemical processes.
No, it's not
normally.
It's not abiotic transformation. It's abiotic removal. So it's adsorption.
Abiotics, yeah, exactly. But I would expect for those processes, the abiotic processes, sorption, desorption. Actually, our experiments absorption is 8 hours, 16, 24, and then we consider that it's enough time for these physicochemical, let's say, processes. So I consider that 48 hours is quite slower kinetics to be an abiotic, whatever, mediated process. No?
It was not the aim of the study.
I know.
I decided 48 hours, I could take samples. And I think that I took sample at 8 hours, but it's not depicted here. At 48 hours to avoid to ensure that I'm detecting the adsorption if it's happening. Just to know, okay, at 48 hours, the removal, it's 90% by adsorption. No. I don't care about the kinetics.
And mainly here is the comparison between hollow and bold. That is very interesting result of this thing, to be the comparison of both things. Okay, more things. I also control my time. So for emerging contaminants, if you compare in general the graphene oxide and the reduced graphene oxide, in general, there are some case studies, it works better the graphene oxide than the reduced graphene oxide, right? Both for kinetics and for the removal efficiency. In general, both are. But this is a little bit contradictory with the methane production, because the methane production, kinetically and yield, is better with the reduced graphene oxide. So I was thinking what could explain this removal, this better removal of the graphene oxide against the reduced graphene oxide. When the sorption is a little bit better with the reduced, the metabolic, let's say, activity I would expect better also with the reduced.
I want to thank this question because it was something that I wanted to say.
Good. It's not prepared, eh? This is not prepared.
Many times, people is studying the removal of pharmaceuticals operating bioreactors at optimum conditions, expecting the microorganisms to do something that they are not prepared to do. In this case, graphene oxide, the only purpose of graphene oxide is, as I mentioned, is to stress the biomass, to put this biomass, this bacteria, in anarchia, in a situation that they have to overexpress other enzymes that in optimum operational conditions, they won't do it. And it's very important to, specifically for the removal of emerging contaminants, is that we cannot study or we cannot spec to promote or to enhance the removal of one very persistent compound working at optimum conditions, because these bacteria will just ignore this pharmaceutical and will focus in easy biodegradable organic matter. Then, what I would suggest is that, okay, graphene oxide works, but it works because it causes the oxidative stress to bacteria. Other compounds is not just graphene oxide. Other compounds, for example, I mentioned in the thesis microaerobic environment. It's a way to stress the biomass and to put it in a tricky situation, so they express other genes promoting the cometabolic degradation of these persistent compounds. So it's, and there is a lot of people working studying the removal of pharmaceutical, but just focusing in this is the best operational conditions for this bacteria, yes, but if you don't put this bacteria under stress, they will ignore this pharmaceutical because they don't uptake energy from them. So that's why we have problems with emerging contaminants, in my opinion.
Yeah, I was going to come to that point, just to my final questions, because if you put all together with this effect of the graphene oxide, no, is one of your objectives on graphene oxide on pharmaceuticals removal, it's like it's an indirect effect of the graphene oxide on the oxidative stress that you place there, and I think it's the future work to go with it. What is this oxidative stress plus some other things? Because it's clear that sorption plays a minor role for some contaminants. But it's why this oxidative stress, also the communities that you identify that are favored, no? The syntrophic bacteria plus the hydrogenotrophic archaea. What is the role of this, let's say, these three things on this emerging, the pharmaceuticals removal on this point?
The main point is that the oxidative stress response causes the production of specific and unspecific enzymes to consume these oxygen reactive species in the media. These enzymes, as I said, are non-specific. Usually some enzymes catalyze one reaction or two reactions with one specific substrate. But since these other kind of enzymes are produced, then can degrade. It's a wild, I don't know how to say it, broad range substrate enzymes. So what I was thinking to say in the previous question is that we have to promote the synthesis of these enzymes to catalyze undesired reaction. Well, in our case, it's desired, but they don't win with this. And usually are oxidoreductase enzymes. That these enzymes catalyze the oxidation, but needs to be reduced at the same time to be functional again. So.
Yeah, it's a cycle. So just my last question. So you selected graphene oxide as a carbon-based material to improve methane production and removal. So how do you place, or how do you have to sell graphene oxide against activated carbon, for example? That is the most common material now, let's say, well, for emerging contaminants and so on, maybe come from Switzerland or whatever. But if you have to defend again you still defend the graphene oxide? Or
yes.
Not anymore? Okay. If you still defend graphene oxide against any other, because you see zeolites for heavy metals, you see not many things, what are you see the three key highlights of graphene oxide against other carbon-based materials, or other materials in general, metal-based or carbon-based, whatever? After what you have seen in these 200 pages of thesis, what will be the main highlights to
Definitely, I need another thing in my to-do list is to do an experiment comparing the main carbon-based materials in one experiment to finally address which of them is the best. Because according to the main results published in the literature, graphene oxide do not increase the methane production. It increases the methane production rate, but the other carbon-based materials produce more methane than in the control. In my opinion, I don't know if this is because these other carbon-based materials can be degraded and use it as carbon source, or if the diet phenomenon can promote the degradation of persistent organic matter present in your substrate. That's why I want to go deep on this. The main characteristics of, well, it's the superconductivity, that it's a graphene-like material, but it has another problem, that there is some studies that report that graphene oxide can be toxic because it's a nanomaterial. And waste management of your sludge containing graphene oxide, I think that the only option is incineration. But regarding the biological degradation of pharmaceuticals, I think, of course, I need to compare it in one single experiment. The oxidative stress, it's a good point that supports that graphene oxide is good for the pharmaceuticals elimination. But we cannot dose continuously graphene oxide in an anaerobic membrane bioreactor because it's super expensive. Definitely. And the main reason that we are using an anaerobic treatment is to produce methane and recover energy. So, to be honest, I would not defend graphene oxide. That's the truth.
Thank you very much. It was a very nice discussion. Now it's time, maybe the directors want to make any comments, question more? No? Any doctor, PhD in the room that wants a further clarification or curiosity? It seems that everything was very clear. Ok, entonces finalizamos la sesión y creo que nos vamos nosotros, ¿no? Vale.
Pues al final sí que ho he disfrutat,
Yo creo que tengo una buena voz, pero... o creía. Nada, decía que, bueno, hemos acabado muy pronto porque no tuvimos mucho que deliberar, la verdad, ya teníamos las cosas claras y simplemente el acto de hoy fue un acto público para manifestar lo que ya creíamos. Entonces, el tribunal considera que has hecho un trabajo excelente, abarcando, como ha dicho, muchos puntos diferentes, técnicos, microbiológicos, contaminantes emergentes. Has dominado el tema y lo has demostrado que es un trabajo que fue tuyo, aunque tuviste directora, supongo, pero has demostrado que dominas el tema y que es un trabajo que lo has hecho tuyo. Conocías los detalles de todos los capítulos en la cabeza, estaba ahí. Eso es algo que has entrado en tu tema de tesis y no es algo, pues no todos acaban con ese dominio del tema. Y entonces la defensa fue estupenda, nos aclaró un montón, la presentación muy didáctica, no solo para expertos en el tema, sino también yo creo que para tu familia. Ahora sabrán lo que son los óxidos de grafeno, si no lo sabían ya. Y las respuestas a las preguntas, pues lo mismo, justificando, pensando, sabiendo que hay cosas que todavía nunca cerramos todo, ¿verdad? Es dificilísimo dejar todo cerrado en una tesis. Eso no significa hacer otra tesis, pero simplemente que alguien siga por ahí. Entonces, por todo ello, el tribunal considera que mereces la máxima calificación de excelente y hasta aquí puedo hablar, porque el resto ya sabes que es súper secreto y alguien te mandará un correo o algo para informarte a ver de lo que pasa en los próximos días. Entonces, yo creo que podemos darle un fuerte aplauso.
Oriol?
No. Direct. Ahora...
Moltes gràcies a tots els que heu vingut, especialment a la cantonada dreta. I també a tots els altres anònims que formeu part de la meva tesi, que m'heu... Bueno, ha sigut una experiència molt guai. Compartir-ho amb tots vosaltres i també amb les meves magnífiques supervisors.